And it is my great pleasure to introduce Diego Porjo-Kress. Diego is, I'm going to tell a little bit about you. He is an associate professor of medicine and neurobiology at Duke University, and I think he is a trailblazer in the field of the gut-brain axis. So his lab's overall goal is to learn how to treat the brain from the gut. And with that, his lab is focused on dissecting the neural circuits that transform signals from food or bacteria in the gut into electrical impulses that modulate behaviors like the desire to eat, what we just all did. He actually discovered a cell in the gut that can guide your food choices. He is also the founder of Gastronauts, which is a global platform that discusses matters of the gut and brain axis. I highly recommend his podcast. And I just actually got back yesterday from a Gastronauts conference in Arizona, which was held in Biosphere 2, which was on where the future of food begins. It was a fabulous meeting, and now I'm really honored to host Diego here. I'm glad he came here. He came here with a very early morning flight, so thank you for being here, Diego.
Thank you so much, Julia. Thank you. How's everybody feeling? You guys awake? So I often say that we're a consequence of two things. The food that you eat and the people that you meet. And this was the first time that I heard, that I had in writing some documentation that I was invited by the one and only Dr. Julia Carlschmidt. And I had actually been thinking of inviting her to a Gastronaut seminar series. And I remember making the not so great choice of coming on March 19, 2020. So finally I'm here after 5 years, But with Julia, COVID didn't hold us back. We have traveled the world. Here is in the middle of the world in Ecuador. In fact, I had the opportunity to organize a symposium with Julia. And I grew up not far from it in the Amazon region of Ecuador. And this is the Curaray River. I recently took this photo, finally. I had seen a similar photo of the Curaray River in National Geographic, and finally I had the chance to take my own photo. And somebody asked me, they said, how is it that you became enthralled by the gut? And then I usually say, where I grew up, even the rivers look like a gut. And then somebody else asked, and how does it feel? And this was a video with a friend in which we were going up the Napo River. And sometimes all of a sudden you're in these dug-out canoes. And this is how it feels. Besides, the drops of water are almost like the shape of an avocado. That's my friend Gustavo, and we got caught up in the Napo River coming from Pantoja all the way to Ecuador some years ago. And I often say that since we are what we eat, eating is one of the most ancient behaviors in code that 600 million years ago, some mischievous cells decided to come together, to live together, to dream together, to eat bigger and better by the division of labor, and that's how multicellular organisms got formed. And it was with the purpose of transforming food into life. And here is a beautiful video that encapsulates this behavior.
A snake's eyes aren't very good, but they can detect movement. So if the hatchling keeps its nerve, it may just avoid detection.
iguana walking on 2 feet. But before I let you see the end, let me tell you what is probably happening inside of the belly of the snake and probably inside of the belly of the iguana too. So as early as the 1900s, we already had a very clear depiction of the mechanisms of digestion and how it is that we converted food into feelings, and in the process we produce feces too. So when we eat a piece of chocolate, that goes into the stomach. It gets baited with some pepsin, rennin, hydrochloric acid, with some juices. And then usually the gatekeeper, Mr. Pylorus, lets the chyme flow into the intestine. And I often say that this is where the magic happens, because literally the gut somehow knows what it is exactly that we need to continue to build life by extracting those sugars, those fats, those proteins, those vitamins. Imagine like all of the complexity. Just a single glass of milk, it has so much complexity in the variety of chemicals, all the way from lipids to like even microbial metabolites that are educating the gut and are interacting with the gut. And partly of this idea that the man as industrial palates, as it was depicted by Fritz Kahn, it rose from the idea that we are a machine and the terminology of the calorie and what is the unit of heat that is required for a motor or an engine to move. And then we translated that into food. And the biggest issue has been that now the communication of the value of food is around this terminology that is made for a machine. But as there was an interesting book in the 1940s that was poking fun at René Descartes and said that the only thing that it cannot be explained by the think, therefore we are, is the ghost in the machine. Because it is the parts that when they come together is that they give rise to something that is more than just like the units. And I think that that's going to be the future of this field, trying to figure out the ghost in the machine, not only the receptors or the cells or the circuitry that are passing one thing to another. And I think that to the point of the calorie, it's very evident that the body does not sense calories. The body senses molecules. And because it does not sense calories, you will see that it's not the same thing as eating just like a 2,000 calorie meal that is made of just carbohydrates than compared to one that is more balanced, right? So, and this, if you extrapolate this, many of you, one of my favorite movies, 1998, I think that this is scene like really depicts the issue.
Do we have a deal, Mr. Reagan?
You know, I know this steak doesn't exist. I know that when I put it in my mouth, the matrix is telling my brain that it is juicy and delicious. After 9 years, you know what I realized? Ignorance is bliss.
Because one thing is that you think about food, and another thing is to feel about food. And since the early 1900s, 1902, we thought that the gut was sensing and reacting to specific nutrients only through hormonal paths. In fact, that's why we call it an axis, because an axis is an imaginary line around which two bodies rotate. Because there had not been description of physical paths that will connect the gut and the brain. And a lot of that has been not only like the investment in the biology, but also the emergence of tools. And since the 1950s, we began studying our sensory systems, and obviously that's very important because it is the senses that gives rise to the reality, and that's how we can process sound, vision, and so on and so forth. And while all of the neuroepithelial circuits, and not only the external ones, but you find there's like 15 or 16 different neuroepithelial systems in the choroid plexus, in the bladder, in the reproductive tract, in the spinal cord, that there are these neuroepithelial cells. And while all of them are considered to be form synaptic connections, direct physical interactions with nerves, the gut was left behind until like 2015. And as I often say, this was a significant gap in knowledge, because even if the gut will be able to send information to the brain did not have the physical means to be able to react to it with the precision of space and time. And that became my lifelong career, and I think that is evolving now into not only trying to understand how it is that the gut guides emotions, but also like taking the stage and hopefully helping especially the young minds that are getting into this field get inspired to explore this, because I think that it is at the intersection between food, the gut, and the brain that we're going to start to see the emergence of new therapies for the future. So when I landed at Duke, in 2010, January 4th, and here's where I usually pay homage to my mentor, Roger Little. Because I had no papers, I had not published anything, and then when I went to interview with him, I guess he saw a little bit of excitement, and he welcomed me. And the other thing that he did is he gave me a choice. And he said, Diego, I have these 2 transgenic GFP mice. And for me, it was like new toys. And he said, I have these cholecystokinin GFP and these peptide YY GFP. Which one do you want? And I said, but just remember, I've been working on cholecystokinin for like 30 years, he said. And I remember my gut reaction was like, PYY GFP. And then he looked at me like, I don't know anything about PYY. And then I said, like, well, neither do I, so we'll learn together, And I often say that obviously that was perhaps bold and naive from my perspective. But I think that there is it's a nice payoff to be able to go into the unknown when you're starting the path in science. And one of the first things that we saw with that tool is that those cells that has been for the long time, they have been described as triangular in shape. Some of those had these very interesting feet that look like primordial neurons. And in fact, here you have this very nice elongation that reminds me of the Sistine Chapel, and it's like where Adam is reaching out to God, and God is reaching out to Adam. And this is actually a nice philosophical thing. I read a very nice article in the New Yorker about that Michelangelo what he was thinking was, is God created man or man created God? Because the painting of the God, if you go and look at it, is actually a depiction of the brain in the background. So one of the first things that I did is trying to track if these cells were contacting the vasculature. And the reason was because the original name that people had given to that system is the enteroendocrine cell, or a gut cell that releases hormones. And that terminology was coined in 1937 by Frederick Feiter, because he was studying the diffuse endocrine organ of the gut. So he called these cells entero for gut and endocrine for the endocrine release. So obviously, the name was saying endocrine, so I went to look for the vasculature. Wrong. The vasculature was not the answer. And here's where I usually emphasize, because one of the biggest gaps that we have in science, and I'm talking to the mentors, I'm talking to the PIs, I'm talking to the students, is that we really don't understand linguistics and we don't understand language. And we often send people inside of the microscope room to go and figure out something new and come back and tell us the story. And the way that we build language is by attaching things that look like things that look like things until we try to describe the whole thing, right? That's how I published my first paper, 2010. Actually, I always highlight this. Received 14 September 2010. Accepted 19 October 2010, 4 weeks later. I think that all I had to do was fix the affiliation. But that was an impact factor of 0.08, I think. That. But that paper gave me, I'm very grateful to this experience because that paper gave me like the beginnings of science, and I started to understand how to put together a figure. And also Roger said, Diego, in this business, publish or perish. We need money for your fellowship, so I think that it will help if you have a publication. And then I got my first F-32. I think that it was largely because Roger knew how to do the magic. He was the ghost in the machine. So eventually, we had to give it a name, and we had to give it a name because we were running out of space in the abstract to be able to call them axon-like pseudopod cytoplasmic processes. So I did a little bit of reading, and I came up with the term neuropods. And then eventually, that term has become adopted for the cells that transmit sensory information very rapidly to the brain. And it didn't take much from the imagination perspective to figure out that these cells were talking to the nervous system. And at the same time, I started to get into the neurosciences. I went to a marine biological laboratory, took the neurobiology course, the Grass Fellowship, and other things. And it has been quite a bit of an adventure. And most importantly, it has been a vehicle to make friends. Because it was a friend that actually suggested this experiment. When I showed up at her office, Nicole Kalakos, who trained with Bob Malenka here several years ago, she said, like, you got to do rabies. You got to show that these cells are actually monosynaptically connected. And the rabies system developed by Ed Callaway and his team, it was based on the rabies virus, which prefers to infect neurons because they have these neurotropic factor receptors. So the rabies virus is very intelligent, hijacks those receptors, and then gets inside of the cells. So this was a leap of faith experiment because we thought, well, the cells look like neurons, may act like neurons, they may quack like neurons, they are electrically excitable. So I remember we did a little bit of magic in there to get enough of the virus because in the brain, 300 nanoliters is enough to do this tracing. But in the gut is an entire tube, remember. And eventually with the help of Professor Fan Guan, now at the MIT, she helped us to figure out the system because she was studying this in the whiskers. So leap of faith experiment, put some virus in. This was a micrograph from 2013, which put in there the virus. And then 7 days later, these discrete cells, this discrete number of a population of cells, were actually labeled by the rabies virus. And something very important in here is that this virus does not have the ability to replicate in a wild-type animal, so therefore you don't see anything else being labeled. And then through, as Nicole calls it, like some rabies gymnastics in which you have to have a specialized mouse that will express the complementary G inside of the targeted cells. Then if we do the same experiment in this G-deficient, glycoprotein G-deficient rabies virus, you can put the rabies virus in there. It will infect these neuropod cells, which express artificially the RAPG gene. And then so the virus will be able to recombine and jump one synapse. So here is the idea. The virus gets inside of the cell, labels the first cell, recombines with RAPG, then jumps one synapse and then loses the G, and it stops there. And that's what we saw. In fact, here you can see some remaining of the GFP labeling these. This is actually a PYY cell labeled with an antibody. And here you can see the nerve fiber. And upstream of that, you see some vagal nodose ganglion neurons labeled by the rabies that was put in the lumen of the colon. I always emphasize that this was a leap of faith experiment. And it was a functional experiment to demonstrate the proof of principle. Eventually, I learned from Ed Callaway that actually it was a miracle that this happened because the rabies depends on the number of synapses in the entry point neuron to be able to replicate. And these cells, as I was chatting with Elaine earlier and with somebody else, these cells only have like 1 or 2 synaptic contacts. And not only the virus was able to get in there and label the vagal nodose neurons, but actually like the terminals upstream of the vagal nodose neurons finish in the nucleus tractus solitarius. So this experiment had two immediate consequences. The first one is that, like the tongue, like the skin, like the eyes, like the lungs, like all of the epithelial surfaces, the gut also has the anatomical machinery to be able to transduce a sense very rapidly up to the brain. But inadvertently, by using a virus, we bump into a potential path for pathogens to crawl up from the lumen of the gut into the brain. And now it's becoming more mainstream that there is quite a bit of neurodegenerative diseases that may begin or may have something to do or may be associated with the intestine. Parkinson's, Alzheimer's, in fact, a prion disease that humans get it by eating tainted cow meat. It comes, from the gut. Eventually, we published some of that work. Around that time, somebody that I hold a lot of respect for scientifically also entered into this field, David Julius, and it has been great to be able to see how this field has been developing in that branch too, because they have started to open a very nice line of investigation into the area of pain. And in fact, Holly and David have recently had a beautiful paper showing that perhaps these cells also give rise to the dichotomy of how it is that IBS or visceral pain symptoms have this dichotomy in males versus females. So back to the nutrients. One of my favorite papers, 1973. Professor Gibbs at Columbia says, opening sentence of that paper says, within 10 minutes of starting to eat, a rat stops eating, grooms for a little bit of time, and then usually sleeps. We define this behavioral sequence as satiety. Its physiological basis is unknown. Imagine, only 52 years ago, we didn't know, the physiological basis of satiety, of how much we eat or why we stop eating or started to eat, right? So they went on to show a few concepts very nicely in the paper. It was one of those classic papers, straight to the point, couple of things supporting that point. And the first one they said is that if you bore a hole in the stomach of the rat will continue to eat at least 30 or 40% more than what the rat normally will eat, because the food needs to get into the intestine for the signals to give rise to the sensation of satiety. The second thing that they go in there and say is that one of those signals is cholecystokinin. And cholecystokinin, the first time that it was described, it was in 1923, and then eventually people were suspecting that may be involved in satiety, and they went on to describe that cholecystokinin perhaps is one of those molecules. The only dichotomy in the issue is that they show in that paper that cholecystokinin takes about 10, 15, 20, and 30 minutes to peak in the bloodstream. So obviously, they are saying that within 10 minutes of the rat starting to eat, she already goes and puts her pajamas on, brushes her teeth, gets in bed, and falls asleep. But the signal of satiety just keeps going, right? So essentially, there is an entire black box in there that we really don't understand. So here is a very nice, beautiful video. And now I have seen other laboratories are starting to adopt this very wonderful methodology that was adapted and propelled forward by Dr. Teresa Lieber at the University of Missouri. So here you have a mouse, starts to eat a liquid meal. 3 seconds, the food is already in the stomach. And in less than 10 seconds, boom, it already gets propelled into the intestine. So well before you have the feeling that your brain is starting to compute what satiety means, the gut has to already at least sniff at what you just ate. And here over the years, I've learned to make this distinction. We shouldn't confuse absorption with detection, with sensory detection, because for sensory detection, the gut just needs to make a little, to be able to get a representation whether you just ingested a friend or a foe, right? And that information perhaps has to be distributed throughout the gut to be able to educate the immune system, also react to the bacteria, and so on and so forth. And in fact, behavioral economists have known that 95% of our decision making is actually visceral in nature. You guys will not let me lie, right? When I go to the grocery store, somehow the grocery store manager, especially the Whole Food guys, they really know this thing. Milk, way in the back with the salmon. You know, like you gotta go with the baby screaming and everything, but somehow you get to the milk, right? And then you come back and you don't want to pay attention to anything else, but somehow like you pick up flowers and bread and like. But by the time that you get to the counter, magically those Kit Kats get into your hand. And by the time that you have gotten to the car, you have eaten like at least half of it or all of them. And as this book, Dan Ariely put it, there are hidden forces that shape our decisions. And we're really, we are predictably irrational. And as early as the 1950s, it was known that food delivered directly into the gut is reinforcing. And meaning that even in the absence of taste, the animal is already able to distinguish sugar from sweetener. So here we adapted this behavior to be able to track. And working with engineer Paulina Anikieva at MIT, we developed a flexible fiber optic to bring optogenetics to the gut, bring light to the gut. Here is, in fact, this little Teletubby friend after a week of the implantation of the ferrule. And moment of truth. In the control light, sugar over sweetener, animal goes to sugar. But if you silence these neuropods, the animal literally becomes blind to the sugars. It cannot distinguish both. And I recommend, I highlight that if you're very interested in the subject, please go to the details in our publications, because a single cell has the ability to distinguish both the glucose from the sweetener part. And then it releases both signaling molecules, ATP versus glutamate. We don't know what ATP does in terms of behavior for the sweetener, but certainly glutamate is reinforcing. And in fact, here we repeated the same experiment, blocking glutamate. And glutamate is the one that gives rise to the behavior. And more recent work, we know that that is actually for fats too. So since sugar and fats are very rewarding, it seems that glutamate is at least one of the main signals that from the gut is controlling your decision making. And this paper was put out in a bioRxiv pretty much in a final form in 2020, but it took 2 years to get it out there. And I think that that's why my way of paying homage to the people that now have quite a bit of stamina to put these pieces of work out there. So just to end, since I was in grad school, I was puzzled by microbes. In fact, I was able to find some microbes inside of the chicken embryo in my dissertation. I never got published that paper because I sent it to +1, and one of the reviewers almost like comes home and slaps me in the face like, don't you know that the gut is sterile before like birth? You know, like, don't send this paper this way. So I never published it. It is in my dissertation. And in 2013, I had read this paper from Elaine, and beautiful paper, very nice story. And towards the end, in like the last paragraphs, I remember starting to wonder, like, what are the cells that these microorganisms are talking to? And as I was sharing with Elaine, that really put us in a path of trying to figure out how it is that microbes work with these sensory systems to be able to guide the brain, and also the brain react to the inputs of the microbes in the absence of the immune system. So since I don't have another 12 years to explain to you the paper, I brought to you a video.
Every organism interprets the world through its senses, its Umwelt. Beyond the familiar senses, our bodies have a hidden sixth sense, a gut sense. This allows us to perceive our internal gut environment, detect nutrients and guide food choices. signals from nutrients in the gut are transmitted via neuropod cells to the vagus nerve, directly to the brain. The colon teems with trillions of microbes forming our gut microbiome. Evidence shows that gut bacteria influence behavior. However, the precise sensors and circuits through which microbes directly influence their host's behavior remain unknown. Neuropod cells are fast transducers of nutrient signals to the brain. They also have receptors that detect microbes, raising the question, could neuropod cells also transmit signals from biota to the brain? Bacteria in the colon move around using their tails, known as flagella. Flagella are composed of various pattern proteins, with a key protein called flagellin, which is more abundant in the lumen when the host feeds. Neuropod cells express the toll-like receptor 5, or TLR5. This receptor recognizes flagellin, a structural protein of the microbial tail flagella. The flagellin protein binds to TLR5 in neuropod cells. Instead of triggering a typical immune response, activation of TLR5 in neuropod cells leads to the release of the neuromodulator PYY. PYY activates the NPY2 receptor on vagal neurons. This sends a signal to the brain, reducing appetite. When we knock out or block these receptors, we have observed that the host becomes overweight due to eating slightly more and over a prolonged period. We discovered that this occurs even in the absence of bacteria. When flagellin is introduced into the lumen of the colon, the animal reduces feeding. We call this sense, at the interface of the biota and the brain, the neurobiotic sense. This neurobiotic sense regulates feeding. Just as organisms rely on their senses to navigate the external world, they also use their gut Umwelt to adjust their behavior.
A nice bedtime story for those who have children. So people usually ask me, what is the future? What is the next thing? I think that if you believe that we're going to be able to get to Mars by 2050, it's going to take us way longer to figure out what is happening inside of us. Because imagine 1, 2, 3, 4, 5, 6, 7, 8 meters, 8.5 meters of gut. And we were, with Julia and other friends, we were just at Gastronauts in Biosphere 2. And there was an absolutely beautiful talk in which they were showing the regionalization of the gut, in which we were, we have learned the duodenum, the jejunum, and with using transcriptomics and genomics, they had this beautiful interposition, not only in mice, but also in humans. And they actually show a photo of like the 6 or 7 meters of gut open of a human. So it's quite a vast space. Imagine what we can do with just like 2-3 inches of nose, right? So just to put a plug in there, often if we think about the future, it's a great way to look at the past to be able to like see where we're headed. Plants. I was a couple of years ago with my family at the botanical gardens of Oxford. And there I found in this medicinal plant garden this very humble sign in which I learned that up to 80% of the medicine worldwide is still provided directly by plants. In fact, if you talk with your grandmother or in some other places, you will still have the tea or the concoction or the ortiga to be able to like heal pain and so on and so forth. 25% of the drugs today are still directly developed from medicinal plants. So the future of medicine, I actually believe in, that is in plants. And partly of that is that I have been exploring the Amazon because it has the largest pharmacopoeia in the planet. And I believe that the decimation of the Amazon is not necessarily us with the machete or the chainsaw or the machine. It's like the lack of empathy and the lack of knowledge or ignorance of the plants, and not in a bad way, but the lack of empathy that doesn't allow us to appreciate what it is that we have. When you get home and you have like one of those monsteras or one of those sansevierias, the snake plants. In South Africa, those are sacred plants that are medicinal. I just learned that from a botanist from South Africa not long ago. And in fact, here are some friends. Here is Cesar from the Kofan tribe. They are the most knowledgeable tribe in the Amazon in terms of medicinal plants. Richard Evan Schultes, director of the Botanical Museum at Harvard, legendary ethnobotanist, describe them as like one of the most knowledgeable people in terms of medicine. And they are like one of the last families that still keep the traditions in there, including the Yahe tradition. And here is my friend Isidro, Taita Isidro as we call him. And obviously you know that each one of those teeth have a story. Because for them, in order to be able to hunt the jaguar, they have to get ahead of the sensory perception of the jaguar, because the jaguar also chews up the sacred plant. And when the jaguar gets into the village, it's very dangerous, so they have to defend themselves. And for that, they have to be 2-3 steps ahead of the jaguar, right? And this is Alex, his son, who recently won actually a Global Sustainability Award because he's fighting to maintain his lands against illegal mining and oil exploration. And here is Rene, who is the brother-in-law of Alex. And I just want to finish saying this has been thanks to the people that I've met. And through some of those people we met, we formed Gastronauts. And Gastronauts, it is in its 10 years. And we just had a blast. So if you want to learn more about this community, especially those that are entering the field, this is a great place. thinkgastronauts.com. The music from the team. You know, I'm here just to entertain for 35 minutes. Hopefully I didn't go over. But I know that you were waiting for this.
A near miraculous escape.
Thank you.
Thank you, Diego. That was fantastic. Questions?
That was an amazing talk, very thought-provoking.
Thank you.
I'm curious about how you think about maybe the timescales and potentially the combinatorial sensing and information flow. Like, you know, I've met chefs who say, you know, sometimes I like to eat dessert first. But of course, from a signaling perspective, it seems like the information flow through this circuit could be very, very different. And of course, in the same way, you eat a 6-hour dinner and your satiety is very different. And I guess, do you have a sense of both how that integration is taking place and how much of it is sort of priority effects versus a sort of integration timescale, if that makes sense?
Yeah, to begin with is not a 0 or 1, right? We're constantly integrated. In fact, your gut is telling you something right now. You know, you pay attention to it. And especially if you start to get thirsty or you start to get hungry, and then the hunger pangs come in. So it's like a continuum, right? It is a continuum. And the second thing is that once we start to eat, this information starts to be computed, but it's also not completely disentangled from the previous meal, right? It's not like the gut is empty, like completely empty. There is always something in there. And that information has to be relayed. And then strictly to your point, we are just beginning to understand how it is that these sensory signals are integrated with the rest of the body. Because, for instance, in patients with autism spectrum disorders or patients with anorexia, it is said that, and there is some strong evidence, that their perception, like what for a neurotypical person will be, that normally you will not pay attention to your gut. For such patients, they will be very hyper aware of what is going on in the gut. In fact, I've seen like some drawings from children with that are developing anorexia, in which they show, like in the painting, they show themselves as like very skinny, but the gut is very bloated. How is that connected to the other senses? Because obviously, like the gut is not acting alone, but the other senses are guiding the experience. More work to be done.
Thank you for that great talk. I'm wondering about the scope and the valence of the toll-like receptors in the gut. What are the range of behaviors that can be triggered by activation of specific toll receptors? Is this like one small subset of the possibilities?
Yes, certainly it is one subset of the possibilities, because as I heard one time, Mike Ehlers, who was a synaptic physiologist, eventually went to industry, said, in academia we tell stories. In industry we produce data, reproducible data, right? Like partly you have to like build the rigs and so on and so forth, right? And this is obviously an archetype. So hopefully other people also like start to wonder like what are not only other receptors possibly in there, like for instance, these cells also express some, in less proportion, other receptors. And when I said in less proportion, it should be taken with a caveat that the receptors in expression are changing. This is very dynamic. We have data in which like fasted versus fed, 2 hours, 1 hour, like you see changes in expression. So these receptors are constantly changing. And then the other part on the behavior, and even if you take the behavior like something as food intake, and the amount of time that the animal eats and how much it eats, right? If you couple that to, I didn't train as an immunologist or as a microbiologist, and then we enter into this world, fascinating world, like just simply something like flagelling is not like there is one flagelling, right? There is like so many different variants. So there is quite a bit of, like just some food for thought in there. We actually got, because of the revisions, we got to think about what actually defines a pathogen. Because at the molecular level, these cells don't know what is a pathogen, what is not, and it's depending also like the salience and the valence, right? Like the amount. So.
Is this neuropod cells and internal neuroendocrine cells, are they one-to-one equal, or one is subpopulation of the other one?
Like the precise phenotyping of like the synapses, what we have seen consistently is that between 2 in 5 of these cells are synaptically connected. And it's very similar to what you see in Merkel cells and other neuroepithelial cells. If you go down into like the specific markers, like we began like studying the neuromodulators, the hormones, the neuropeptides, right? PYY, CCK. And in fact, like we know that 4 out of 10 of the CCK cells also express glutamate, but there's not a full overlap. In terms of like, how do I see the picture? I think that there's at least 4, and I don't like to call these families or types, because we have gotten into the habit of describing these cells by one gene that they express. And as I often say, that will be like trying to take your mother, compress it into a pill, take a pill, and then feel like you have your mother. You know what I mean? Like it's, one receptor doesn't make the whole cell, right? So I try to be cautious when I say this about the families. But I do think that there are some cells that like send signals, receive signals, perhaps do both, and there seems to be one reservoir of neural stem cells, like some cells that can regenerate in the villus, in the shaft of the villus, of these neuroendocrine cells.
But then, so how do they distinguish like a pain signal versus a satiety signal?
Receptors and signals. They express different receptors, and they have different signals. So they will express the TRP channels, and they will secrete serotonin. And even on the serotonin story, like enkephalins, for instance, are expressed within the cells, and it has not been explored yet.
But how do the cell know?
We can continue to chat after. If anybody has any final question, because I know that.
Hi, thank you very much for the talk. It was amazing. Maybe it's a naive question because I'm not too much in the field, but I was intrigued by the part that you showed the relationship between the bacteria and the brain. And so can we say that from an evolutionary perspective, the bacteria that have been living with us, they kind of hijack us and can affect how we want to eat, how much we want to eat, and what kind of things we want to eat?
So to the naive question, naive questions bring fresh answers, you know. We learned to coexist, because these things go like millions of years ago, so we needed to have this coexistence, right, number one. And then just to put it in very general terms, because this is obviously a very general audience, as I often say, you know, we have one thing in common with the bacteria is that we both need to eat. But the poor little guys don't have the feet to get to the store, right? They need us to get to the store and then get enough food for both of us, like the specific proteins, the specific fibers, highly digestible fibers. So they have to rely on us to be able to get that. But at the same time, we also rely on them to be able to get some vitamins and some specific molecules that help us to coexist, right? So hopefully that gave you an idea. Thank you.
Thank you.